Right. So this is where university really starts, right? So we, we're going to be 6 people presenting in this session, 10 minutes each. Unfortunately, couple of 3 people haven't been able to attend today because of various personal reasons. So you'll be subjected to my voice 3 times than originally it was intended to. But there you go. So we'll probably just introduce ourselves as and when they. Is it this? Yeah. So let's start with why do we need to do research in Kabuki syndrome? So we've already discussed during the clinical time lots of questions which still remain to be answered. And essentially, if you boil down these questions, they would fall into these categories. So we still need to improve diagnosis, and I'll explain how and why we need that. We need to understand the natural history. So for example, Kate, you mentioned about skiing, and you mentioned about skiing. Now, whether that's part of the natural history or is it because of growth hormone, we don't know, right? And that's really important because until you understand that natural history, you cannot really do the best for your patients in terms of their management, screening, treatment, etc. We also need to understand disease mechanisms. And that is important because if we are thinking about new treatments and therapies, you really need to find out, okay, how does this KMT2D genetic chain affects the cells? How does it affect the organs? And how does it affect a human being? So that disease mechanism needs to understand. And then if you do that, then hopefully you can identify potential new therapies and treatments. And once you do that, we need to conduct clinical trials. And if we accomplish all of this, then you need to set it up in a clinical context to make sure that patients and families are benefiting with that. But how do we do that, right? So it seems actually pretty difficult to do all of this. And you can really do that if you've got the infrastructure. So that's really important. I think we're very lucky here in Manchester to have the right infrastructure. Oh, sorry. So in terms of the infrastructure, we've already talked about the genetics department here. We've recently been funded by NHS England for a genomics network of excellence for rare diseases. And that's David Gokhale, one of my colleagues, who is one of the co-leads for the network of excellence. And that is really focused around this question of improving diagnosis in patients. So we've got the right set of people and equipment to help with that. Then we have a Manchester Biomedical Research Centre, which is funded by NIHR, which is the main funding body for clinical research in the UK. And there are several BRCs across the country, and these are each BRC has its own emphasis or a set of emphasis for their research. For the Manchester BRC, I think we're the only theme in the country, we're the only BRC in the country that has a dedicated theme for rare conditions. So that really tells how important we think this whole area of research is. Recently, MRC, which is the premier medical research funding agency, they have set up what's called a UK rare disease platform. So these are research platforms, and within that, they have approved funding for epigenomics of rare disorders. So Kabuki syndrome is it's a genetic disorder which changes the epigenome of the individual. So that's a big investment from MRC, and that confidence from the MRC tells us that we're also on the right path and that it's recognized. And in the UK, this is really being given a lot of importance. So all of these are new initiatives, and hopefully some of the talks that we'll give will tell you how we were utilizing this infrastructure. And this is Professor Bill Newman, who co-leads the rare conditions team in the BRC. This is Professor Albert Besson. He is in Exeter. So Bill is in Manchester. Albert is in Exeter. He was going to come to this event today. Unfortunately, he's not able to attend. So I'll present his slides. And this is Professor Sue Kimber, who works with us as part of the BRC and for the EpiGen Rare platform as well. And these two pieces of infrastructure, they are about improving natural history, understanding mechanisms, and identifying treatments. So all of this is focused on the middle bit. And then finally, we have an IHR-funded Manchester Clinical Research Facility. So there are not that many facilities in the country where you can do advanced therapies, new therapies for rare conditions. This is Professor Simon Jones, who heads the Children's Clinical Research Facility in Manchester. And Professor Jones and the team at the CRF here, they've done gene therapies for brain disorders, for eye disorders, for a number of other metabolic disorders. So overall, I think it's about 60 advanced medical therapies that have been conducted, clinical trials for which have been conducted in the Manchester CRF. So really, we have everything in place. We just need to make sure that this is all aligned and works the best for this community. It should be for Kabuki syndrome. So hopefully, we'll be able to do that going forward in the future. I'll keep on mentioning this as well, because without money, actually, nothing really happens. So you really need the infrastructure and you need the money. But the third and the most important component is, I'll come on to that later. There was one more thing. So what we try to do with Manchester Rare Condition Center is try and coordinate all of that together, because rather than having these several disjointed pieces of infrastructure, you really need to have a joined up thinking so that what we are doing here relates to what we are doing here. Okay, so now coming on to the third and perhaps the most important thing is young researchers, right? So you really need people who can do the work, and and they're training to be really the best at what they can do, and really hopefully preparing for next 10, 15, 20, 25 years or so. Long career and a few. Right? So what I'll do is all of these individuals, they've done some work or they're doing some work on Kabuki syndrome. So it's really what we've done over the last 7, 8 years and what we will be doing over the next 3, 4 years. So what I'll try and do is start with one person at a time. So those who are here, they'll do their talking for themselves. Those who are not, talk on their behalf. So this is Victor Fondes, Dr. Victor Fondes. So he's a geneticist from Chile, and he came to Manchester funded by the Chilean government. So they have a scheme called CONICET, and he was working with us on basically focusing on this topic, how to improve diagnosis for Kabuki syndrome. And please do tell me if I'm too slow or too fast. Just it's a very long presentation, right? No, don't worry. So first of all, what is the challenge? What is the challenge of improving the diagnosis? I think in this room, probably to most people, that doesn't seem like a challenge because you've already had the diagnosis, so you want to go to the next step. But I think as a community, we're also working for those people who haven't yet got the diagnosis, right? So this is really important because until you get the diagnosis, rest of it can just not fall in place at all. So this is a cartoon of KMT2D. So if you remember from my morning's presentation, this is a piece of string of DNA that makes a protein, which is called KMT2D. For most patients with Kabuki syndrome, there is a change here in the DNA. So that little star there is a change, which results in this protein not being able to synthesize in the cells. So there's a stop signal here rather than for the protein to continue being synthesized. So that's relatively easy to interpret. You see this change, you know this is what it is predicted to do, and you can give the diagnosis. However, there's also a subset of patients, so that's about 10 to 15% of patients, in whom this signal is different. So it doesn't cause a stop change, but it just causes a tiny little change in the protein sequence of KMT2D. So these changes are called missense changes. So this is a stop change, and this is a missense change. Stop changes are easy to interpret, but missense changes are much more challenging to interpret because the change is so tiny. So what Victor did was he did a very large study where he compiled all the genetic changes known, all the missense changes in KMT2D that are known in the population or that have been described in patients with Kabuki syndrome. And what he showed was that there are different regions in Kabuki syndrome, in the KMT2D gene, where you see, if you see a missense change, then it's more likely to be disease-causing. And there are certain regions where if you see a change, then that you would not see a change in Kabuki syndrome. So there's a big piece of work that probably Victor spent about a couple of years doing this, and I've just described it in 2 minutes. But the point is that all we need to remember is genetic testing is difficult to do and difficult to interpret, and actually someone, that Victor here, he has actually contributed quite a lot to improving the diagnosis of Kabuki syndrome. So that's one of the ways in which we need to improve the diagnosis with Kabuki syndrome. There's also other issues in diagnosis. So in this particular figure, 3 of these 5 individuals, they were given a diagnosis of Kabuki syndrome by their clinician. So they were said, you have Kabuki syndrome, and then genetic testing was done. So if you remember my library analogy, so it was done. People looked at KMT2D gene in that library, but there was no change. So these were individuals who were thought to have Kabuki syndrome, but they didn't have KMT2D or KDM6A mutation. So Victor studied a cohort of these individuals, and he found changes in a gene called EIF5A. So that ended up being described as a new condition that was previously not recognized. So obviously it directly helps these patients and several dozens of patients more to achieve this diagnosis. Otherwise, previously, they would not receive appropriate medical management. So you might think, okay, they don't really have Kabuki syndrome. They've got something like Kabuki syndrome. Why are you talking about this at a Kabuki UK conference, right? But I think it's really important because it tells us about something about the fundamental biology of the condition as well. So what we also found when we did more research into this particular gene is that this gene is required for synthesis of KMT2D in the body. So when I go back, so there is this piece of DNA that needs to make this protein, but it's in the cell, it's not a simple arrow. There's hundreds of factors which are required to convert this DNA into this protein. And EIF5A is one of those important factors. So if you're missing, even though KMT2D sequence is fine, the KMT2D protein is still perhaps not being produced as it should be. So it is really telling us something fundamental, which hopefully we can understand the gene better and maybe even develop some treatments for Kabuki syndrome going forward in future. So that improves diagnosis as well. And then as I said, there's a cohort of patients who are Kabuki-like, but we don't have a diagnosis. So we've solved some of those patients, but there are several patients who still remain unsolved. So this is Dr. Adam Jackson. We did his PhD in Manchester, funded by European Council, and he's found a few other genes, mutations in which cause Kabuki syndrome. So we're still continuing to work on that, and hopefully in 2024, fingers crossed, we'll see those papers come out as well. But I think some of these findings are really very exciting, and it's probably changing, it might even result in changing the way in some of the way in which we think about Kabuki syndrome. So I'm glad that Adam's now finished his PhD and he's decided to stay on in Manchester. And now he's funded by NIHR to do more research really in this area. So he's probably going to take this as his sort of professional expertise, that he's another one of us who wants to improve lives for people with Kabuki and related disorders. So that's really a very good, I'm very happy with that. So Victor's gone back to Chile and he's set up Kabuki clinics there. So again, hopefully what we're doing here, that leads to things getting better all around the world. Then I'm going to talk about Sarah. So Sarah is a scientist, PhD scientist who's been working with us for a few years. And she was funded by a charity called New Life. And the Scales family, who I mentioned in my clinical talk, they are very kind and they supported some of Sarah's research as well. And I'm going to take completely different track now. So I talked about improving diagnosis through understanding KMT2D mutations better. I talked about patients who look like Kabuki, but they don't have KMT2D or KDM6A mutation. And now I'm going to talk about patients who have KMT2D mutations, but don't have Kabuki. So that's really sort of flipping the things on its head. So you might need to concentrate a little bit on this. So there are certain changes. I kind of almost gave it as the gospel truth that if you've got a KMT2D mutation, then you have Kabuki syndrome. But there are certain changes that we identified where patients had KMT2D mutations, but they didn't have Kabuki. So how does that work? So without, again, going too much into the genetics and the science of it, here, first of all, we needed to prove that they have mutations in KMT2D, which we did. Then we needed to prove that they don't have Kabuki syndrome. So the clinical features that these patients had were almost opposite of Kabuki syndrome. So for example, I'm sure you all know that in Kabuki syndrome, patients often have quite prominent ears, right? So quite sort of big ears, sometimes slightly what you call lop ears. And I'm looking at my ENT colleague for that. But as you can see in this patient, perhaps in this little baby, the external ears are completely absent. In here, the external ear is really, really tiny. So there are certain features which are absolutely opposite of what you get in Kabuki syndrome. These children also, they don't have any learning problems. So they have quite more severe malformations, different kind of malformations, but no learning problems either. So motor delay, intellectual problems, and kidney problems are missing. So that told us that there's something else that these mutations are doing than Kabuki syndrome. And why this is important to know, because if you don't recognize this, then you might see, okay, KMT2D mutation, you've got Kabuki syndrome, and you get a management plan which is for Kabuki syndrome, whereas you don't have Kabuki syndrome, you need something completely different. So therefore, the point about improving diagnosis is it works in both the directions. I will not describe that right now. So Victor was a super productive PhD student. I think he wrote 8 or 9 papers in his 3 years, and all in top journals. So the EIF 5A journal paper that we wrote that was published in Nature. So and then he also did something super useful, which I think was slightly ignored before he did this paper. So he also not just worked on improving diagnosis, he's a doctor, and he also wanted to improve the understanding of the natural history of the condition. So in this particular project, what we did was we focused on the rarer type 2 X-linked Kabuki syndrome. And I know that there are a few people who have family history of X-linked Kabuki syndrome. Can I just ask how many? So quite a few. So probably just a shot like this would tell us maybe more than 5% that's quoted in the literature. It's a very sort of a non-scientific poll, but the problem is before this paper, if you looked at the medical literature, all the medical literature really, I would say 95, 96% of it was from type 1 Kabuki syndrome, right? So Victor collected data from over 100 families, I think, and 100 patients, and we put together their clinical features, their genetic changes to try and understand the pattern of problems in type 2 Kabuki syndrome. And what we could show here, these are very complicated, detailed graphs, but I'll just give you the key take-home messages. Essentially, in some ways expected, boys with type 2 Kabuki syndrome are much more severely affected. So, for example, if you look at weight here, so blue are boys, so lower means they have lower weight, pink are girls, and as you can see, the weight is more in girls, so they're not as delayed with their growth as boys are. So that's objective evidence that boys are more severely affected, but that's only for type 2 Kabuki syndrome, so this does not apply to type 1 Kabuki syndrome. Just wanted to make that clear. Plus, this is just the frequency of individual features, and literally for every clinical feature, you would see that the blue bars are higher than the pink bars. So boys are more frequently affected by certain clinical features in type 2 Kabuki syndrome. So that understanding directly improves the understanding of disease and therefore hopefully translates into better management of individuals who are affected. I think here's the next one here, Aoife. So please come through.